students getting a B and a C. So ours was like this, which there's a chasm here. We had no C's and a lot of A's and B's and a lot of D's and below. So there's two things that I've kind of been thinking of, is that one, maybe it's the background, like background, science background at all, or just remembering from a previous class. A lot of times also it will be just study habits. So if you guys in this realm are really wanting to get an A or a B, then please see me after the lecture, and then I'll let some people go. So if you are like, I am not doing as well as I want to be doing, help, then please see me after lecture and we'll do it together, okay? And then we'll make a plan, and we're like, okay, let's, step 1, step 2, let's make a plan together to make it happen. So, yeah, that's what I wanted to say about that. So if you're doing well, so I'm super pumped on how the midterm grades went. And just as a little FYI, it's half of what the final is gonna be. So that was 50 points, the final is gonna be 100 points. And then the final, we're adding all the eukaryotic organisms that we're gonna talk about today. So we'll have another whole slew of organisms for note cards and all that kind of stuff. But I will have higher expectations by the time we get to the final too. You have to underline your organisms. You do need to make a stop codon. You do need to know some of those things that we've kind of covered. And just, you know, to make sure, if you feel like some of the fundamentals, like what is peptidoglycan? What is the difference between a gram positive and a gram negative? If you're missing some of those fundamentals, now is the time to deal with it, not before the final, because those fundamentals, they're just, you know, we're gonna forget about bacteria, except for in the lab. In the lecture, we're gonna forget about them. And then we're gonna come back to the final and be like, oh my gosh, now I'm supposed to learn about bacteria while we have all these other organisms that we've discussed. So just make sure you take the time right now to go back through the fundamentals with me, with Dr. Rosevier, with Dr. Welsh, and we'll power it out. But it does take time, you know, studying just takes a lot of time. The difference between me and another student is that I have to take more time than everybody else. It just takes me longer to study than it does everyone else. But the difference between me and the student sitting next to me is that I'm willing to put in twice the hours as everybody else. So I'm willing to get up at 4 in the morning and sit with my coffee in the quiet with my classical music on and just power, study. Take a break, refresh, wash my face, take a shower, do a little few things, sit down, another 2 hours. Get up, move around, come back, another 2 hours. Like, I will put in the hours for any class. So if you guys are willing to put in the hours and you're willing to put in the time, like, I will cartwheel all day long for you to help you and sit down with you and make sure that I make your success a priority to me as well. Because I want everyone to get an A in this class. That would be ideal for me. So if you are willing to put in the time, I'm willing to put in the time, we can work on it. Okay, so viruses, we're done with viruses now. It's a good idea not to forget the whole bacteria, viruses, of everything that we went over. It's a good idea to recap some of this, keep it in the forefront, keep your note cards, because those note cards are gonna come up again. For the final, you won't have a list to go through. So the final, you're gonna have to pull, like what, oh, that's Staphylococcus aureus. Oh, that's Pseudomonas aeruginosa. So it's a good idea to just keep flipping through those cards and keep those with you, because we don't really have that many note cards left. And when you look at bacteria and viruses, those are the two trickiest ones in my mind. Then we start looking at protozoa, which are so different. And we look at fungi, which are so different, and helminths, which are so different. So you'll be able to kind of separate those. Bacteria and viruses are the most tricky in my mind. I don't know, you'll have to tell me what you think later on. Okay, so we're moving on. So today we're just gonna do like an introduction to prokaryotes, or to eukaryotic organisms. So we did our prokaryotes. So we did our bacteria and our archaea. Let me we did our bacteria, our archaea, and then don't forget over here are our weird little viruses. And so over here are our weird little viruses, and they don't belong on our phylogenetic tree of life because they're not living, right? So we've done prokaryotes, which don't have a nucleus, which don't have a membrane-bound organelle. Bacteria are different than archaea because bacteria have, what do bacteria have? Peptidoglycan. Archaea look just like bacteria. What type of ribosomes do these ones have?
70S.
So these ones have different types of ribosomes. So bacteria and archaea are very simple, but they're also very effective and efficient, and they've been around for millions of years longer than we have. So just because of their simplicity does not mean that they're not effective. So bacteria and archaea we're done with that section. Okay, so let's move on to eukaryotes. So we're going to talk a lot about some really funky organisms, which I think are really cool. I do love when we talk about our protozoa section. Fungi bother me a little bit because I don't really like where we mesh between the plant world and the animal world. They kind of rack my mind a little bit, but hopefully I tried to simplify it for you. And then helminths are just disgusting, so bear with me when we get to that part. But they're just crazy diverse and so weird. Some of the animals that you think would be animals, or some of the protozoa or helminths or fungi that you think would be microorganisms or that they're living, they're just extremely strange and cool. Okay, so we talked about bacteria and archaea. We talked about some of the shapes and arrangements, what they look like on the outside, what they look like on the inside, some of their toxic, pathogenic qualities, and just some characteristics about them. We talked about how to test for them, and we got through our bacteria and archaea section. Our viruses, we talked about some of our shapes of viruses. We talked about viruses getting into and out of cells and that they live inside of cells. We talked about how the different DNA and RNA makes them different. DNA viruses will mutate less rapidly than RNA viruses in general. They are extremely successful on the planet, and yeah, so a lot of viruses make us sick. We use vaccines to help prevent viruses because we don't have a lot of antivirals. Yeah?
Were we going to cover prions anymore?
Let me see if we have a little bit of time at the end.
Okay.
I wanted to see if we could get through eukaryotes, but it is a good idea for us to talk about prions, but prions, it'll take us like 7 minutes to get through, so
Okay.
let's see if we have time. Okay, so eukaryotes, the reason that eukaryotes I think are cool in my mind is one, it's almost like looking back on evolution. Like if you get to look through bacteria and look through archaea and look through protozoa, it's like, you know how people say there is no half chimpanzee, half human. So how could we have evolved? Like where did the middleman go? Where did that evolutionary branch go? Well, there's reasons that things split off and one is more successful than the other, or they're both successful in their own right and they split off evolutionarily. Well, this is what we're kind of looking at as we go along. Is like, well, what? There was a bacteria, and then maybe one was engulfed by another one, and then we get complexity, and so we get to like see evolution as we move through. So I think it's really cool. Protozoa are crazy diverse. They're everywhere in the world, but mostly protozoa. What's different about them is that a lot of times they have to be aquatic. So they have to live in aqueous environments and float around and be able to move around because they're bigger than bacteria and they have to be able to find food. So we're going to talk a ton about protozoa and how cool they are. We're going to talk a lot about fungi. These are the weird ones that kind of branch the plant life, animal life divide. But there are a lot of pathogenic fungal organisms like black mold. We'll talk about like the mushrooms in the yards where it's you tell kids like don't eat that or you'll die. You know, like so we'll talk about what is poisonous about them, and we'll talk about penicillium and how we'll do a little antibiotic review when we get to that section. And we'll talk about how beer is made and bread is made, because that's all fungal organisms. And then we'll talk about helminths, which are gnarly, and they are way more common than you would think or want to know. And yeah, they're mostly intestinal. They can be transmitted through feces frequently, and they're in a lot of places in the world. So we'll learn a lot about those too, unfortunately. So this is a journey to the microcosmos. I want to show you guys this video, but I'm going to move on for a second, and we'll see if we have time at the end, because there's another video that I really want to show you and make sure that we don't run out of time. Okay, so we're going to talk about two main types of fungi, which they're two broad categories. Protozoa, there's 4 broad categories of protozoa we're going to talk about, and then we're going to talk about helminths. But one thing I wanted to bring up about eukaryotes, we'll go through all of these about eukaryotes, but one thing, you know, here, multicellularity or unicellularity. This is where stuff gets weird, because even a professor one time asked me, well, how does it work if there's one cell, like one of our cells, versus a single-celled organism? Like, how does that work? A professor asked me that. So I want you guys to know, like, this is all information that the general public just generally does not know. And so when we're talking about eukaryotes and we move up into complexity, it's just strange. It's like you go from viruses, these tiny, tiny things that maybe are living or maybe not, then we go up to bacteria, which are a little bit bigger. We think they're living, they're moving around, they're doing their thing. And then we move up to protozoa. And so protozoa, it's like one cell that's moving and living around. So if you have a person and you have one skin cell, like this is like a keratinocyte, I shouldn't draw, it looks like an eyeball. So here's one skin cell. Single squamous epithelium for any of you. So there's one keratinocyte, one skin cell. That one skin cell that's laying on the table, like, can't function on its own. It can't go eat, it can't go live, it can't move around, it can't, but a sperm can. A sperm can move around. It does metabolize on its own. So we are multicellular organisms where all of our cells have to work together, and one cell cannot go live on its own, but we're still a eukaryote. But there's these other things that are called protozoa that kind of introduce us to what a eukaryote is, where they are one cell, and that one cell can go and live and function on its own. So even though we all belong in this category, it's really weird that it's almost like we're moving up in complexity, right? Because bacteria can live on their own, protozoa can live on their own, one cell cannot live on its own, but we have to have organs. They have to work together. So it's almost like it's almost like if you imagined us as one big cell, for example, and all of our organs were organelles, that all of our organs were a functioning unit to make us whole. That's what it is, basically. It's like all of our organs are functioning units, where a protozoa has organelles, and in those organelles, they're all functioning units, but that cell can live on its own, and our cells can't. We became more complex, where cells needed to divide and conquer, where they had to divide up the labor to make us more complex as we became more multicellular. So I don't know, I just kind of went on a tangent, but it's so interesting to me how it works in complexity, and I think it's cool because you can kind of see evolution as it goes along. Okay, so these are the organisms that we're going to talk about. Actually, we're not going to talk about algae. Even I did that the first semester, and I loved our algae section. but we ran out of time to do that. But we did talk about viruses, we talked about bacteria, archaea, we're not gonna talk about algae. We are gonna talk about fungi, we're not gonna talk about plants, we are gonna talk about protozoa, we are gonna talk about helminths, but we're not gonna talk about animals. So just if this little chart helps you, I made it myself, so it's not like crazy helpful, but just so you can kind of categorize and like start seeing like where things belong. Like what are we talking about? Are we talking about algae? Are we talking about phytoplankton or zooplankton? Are we talking about you know, like the smudge that grows on rocks? Are we talking, what are we talking about? So what we do for this class, even though there are some microorganisms that we don't really talk about in this class, the reason we don't talk about some of the microorganisms is because they're not pathogenic. So we focus on the pathogens for this class because it's related to health. But there are some microorganisms we don't really talk about, but then some, as you move up in multicellularity, they kind of move out of our micro branch, except for helminths is our only weird one. So helminths, they put them in our group as microbiologists because they're pathogenic and because they can be microscopic, depending on the type. Okay, so just to confuse you all. So here's our prokaryotes, here's our eukaryotes. Now we're on our eukaryotic organisms. Here's some of the main differences between bacteria, archaea, and eukaryotes. So you can go through those and we'll talk about those as we move along. So let's talk about eukaryotic cells. So remember we talked about before nut and after nut. So now we know eukaryotes actually have a nucleus. So if you compare a bacteria with the nucleolus with the DNA just randomly in there, ribosomes, what else do they have? Plasmids, apilus, they can have apilus, fimbriae, they have peptidoglycan, all that kind of stuff. So then we move up and eukaryotic single cells are bigger in size, and we're moving way up in complexity. So now we have organelles. I'll go through these. But now what's happened is we have moved up in complexity, and we have all of these different structures that do different things. So bacteria, the way that they produce ATP is because their electron transport chain is on that inner membrane, on that cytoplasmic membrane. And they can undergo binary fission reproduction. They do have enzymes, they have some vacuole pockets and things like that. But now we're moving up in complexity, and now we get into one eukaryotic cell that has crazy division of labor. Okay, so here is a generic eukaryotic cell. So sometimes eukaryotic cells look like this, sometimes they look like this, sometimes they look like this, sometimes they look like this. They have different shapes, but basically they all are just one generic cell. So I tell my other class that when I teach Bio 100 Anatomy and Physiology. When you learn about cytology, you learn about the study of cells, professors draw it like this, and books show you like that. But human cells come in the shape of columnar cells or flat cells or neurons that can be meters and meters long, or they come in sperm, or like astrocytes, or these weird cells in the brain. So all of our cells come in different shapes, but just broadly you always see it as a circle, a generic circle. Okay, tons of complexity compared to a prokaryote. So here we are with our ribosome. So we talked about our 70S, which are 50 and our 30S subunits. Now we're talking about a eukaryote, a 60 and a 40S subunit. So Remember 60 and 40, that doesn't equal 80, but those are the Svedberg units that it's centrifuged, and that's where the centrifugation, where the line is, where it splits out the pieces of the protein. Okay, so here's all of our organelles. So what I'm gonna have you guys do actually is read that and memorize it. I'm gonna turn it off, and you guys are gonna try to remember that. So try to, it's probably too small for you to identify. But look through those, see if you can draw them, and then we are going to talk about their function, the organelles and their function. So maybe you, two of you can get together, and two of you can get together. You two can get together. And maybe you three can get together. And then you two can get together. Maybe let's have you, so Dr. Otto, you three, go sorry. And then you three right there. And then I'll have.
It's gonna get yellow. Wait, when we throw Ashley?
Okay, ready?
Unless you want to.
Alright.
So maybe you draw this on first. Draw it down.
Here's a pen. So Dr. Lal, if you can move over, that'd be awesome.
Maybe something like.
Tell me if your pen doesn't work, I'll bring you a new one.
Alright, go ahead.
We're gonna draw this? Here, you go.
You feel really confident with this, great. If you don't, I understand.
Okay, we're going to so this will be.
Okay, 3, 2, 1. See if you can draw a human cell or any kind of eukaryote.
Well, I guess we should start with that.
Aw. Start with the ones that you remember. There's smooth as a present, particular one. There's the rough as a present.
No, please don't.
Oh, I'm
That would take
I was
forever.
like, yeah. Let's do it.
Drew the cutout. Oh, okay. Just gave this lesson on my feet.
Yeah, that's fine.
Oh, no, I'm sorry. The artist has not. No, that's ribosomes and this. Actually, we could do actually probably your They're actually in there. Yeah, the ribosomes.
Yeah, it's kind of the same thing, but just one of them has like a little bit of
What phase is our cells in?
Yes, lysosomes are possible.
That's
They were like,
There's gonna be stuff with vacuoles. Yeah, they're just little packets of stuff. Some of them have like digestive enzymes inside of them. They're just little circles.
Okay, everybody ready? Okay, let's flip it over and do it together, and then we'll flip it over, do it, let's draw them all together, and then you can flip it back over and make some adjustments to yours. Okay, so let's just name, and then we're going to go name it again and do function as well. So we'll do structure first, then structure and function. Okay, so what's this big guy right here? That's the nucleus with all of the chromosomal DNA. Then what's this little guy?
Nucleolus.
So this is called a nucleolus. So we have a special little nucleus inside of the nucleus. That special little nucleus carries special DNA. That special DNA codes for what? Does anybody know? What that DNA codes for? It codes for ribosomes. So that's how important ribosomes are. Ribosomes are DNA, RNA, and amino acids are what make human beings. That's what makes us. Is the code that codes for the RNA, that codes for the protein, is what governs everything we do and everything we are. So we have a special little pocket for our ribosomal DNA. That's the nucleolus. Okay, the envelope here, the nuclear envelope fuses with this little, what it is like a set of chambers, almost like another long squirrely membrane. If it's covered in ribosomes, what is it called?
The rough endoplasmic.
The rough endoplasmic reticulum. So make sure to draw this as we go along. Nucleus, nucleolus, rough endoplasmic reticulum, and that also moves out to the smooth endoplasmic reticulum. So it doesn't have any ribosomes on it. Now it's smooth. Okay, so now one more envelope-looking, membrane-looking thing is the Golgi. So this is the Golgi body or Golgi apparatus. I drew it small, but it's another large set of membranes and vesicles. And we'll talk about what its function is in a minute, but that's the Golgi. These little polka dots everywhere, those are ribosomes. There's ribosomes everywhere. These little bean shapes are what?
Mitochondria.
Mitochondria. So the mitochondria, some cells like skeletal muscle have tons of mitochondria. Some cells like skin cells, not as many. And then if it looks like water in here, what's the water?
Cytoplasm.
Cytoplasm. It's more viscous than water, so it's a little bit more thick and like goopy than water is. But there's the cytoplasm on the inside. And then what are these little magnet looking things? I always kind of draw them like
Centrioles.
that. Centrioles. Yeah, so centrioles, we'll talk about what those do, but just draw them, stay with me. This little crosshatch pattern, the crosshatch pattern, remember this is a three-dimensional structure. If this is a cell that actually is shaped like a sphere, then that structure will help keep the three-dimensional shape. What is that? Cytoskeleton. So cyto means cell, cytoskeleton is cell skeleton. So those are proteins that make the three-dimensional shape of the cell. And then what else am I missing? So these little pockets, these little bubbles I don't usually have you guys differentiate between all the different vesicles, vacuoles, peroxisomes, lysosomes. Just broadly you can call them vesicles, broadly you can call them vacuoles, it's fine with me. We have tons, you know, we can name them, but actually we have hundreds of different kinds. So to name them is a little bit, you know, but they have different jobs. And so we'll go through some of the different jobs that they do later on, but you can broadly just call them vacuoles or whatever you want to call them. And then our last and very important piece is this piece. What's this piece? Yes, the cytoplasmic membrane that is made up of a phospholipid bilayer. So when you zoom in, it's phosphates. We just draw them as circles, but it's a phosphorus with oxygens. It's PO4 3- is a phosphate. And then here are lipids, which are carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, are lipids. So lipids on the way down, and then phosphates on the outside. So remember, these lipids are hydrophobic. That means they don't like water. Water phobic phobia. So they don't like water. So if you drop a bunch of phospholipids that we've made in a lab into water, they'll immediately form a bilayer because they turn in towards each other. So these phosphates like water, so they're hydrophilic. These lipids are hydrophobic. So now we have our phospholipid bilayer that surrounds the outside of our cells. Or just our eukaryotic cells, that's our one little protozoa swimming around, has all the same things that our cells have in them. A lot of the same things. Okay, great, now let's go over function really quickly. Wait, flip it over and draw whatever you missed. Go over together with your partner whatever you missed.
Yeah, we did business cytoskeletons, but I feel like we get extra credit for differentiating stuff.
Did you guys miss anything? You got it all? Awesome. Bless you. Pores? No.
Yeah.
No, we're not. So we don't really talk about, we only talk about like receptors and channels and things like that when we get to the immune system. But not really. So I was actually, that's one of my favorite things to talk about. Like, cell receptors, yeah. I love talking about like transcription factors and alterations of the genome. I think that's so interesting. But
Okay, yeah, that's good.
so cool, yeah. Yes, yeah. If you ever get a chance to take a cell bio class, yeah, it's so cool. Or cancer biology, it was one of my favorite classes. Immunology is a great class, yeah. Okay, let's move on. Okay, so here we go. The nucleus. The function of the nucleus is to, let me find a good place to write this down. This is not an ideal place to write it, but stay with me. Okay, the function of the nucleus is to protect DNA. The function of the nucleolus is what?
Protect ribosomal DNA?
Ribosomal DNA. Then we have rough endoplasmic reticulum, RER. So if it's covered in ribosomes, what do ribosomes do?
Making protein.
Ribosomes make proteins, manufacture proteins. So rough ER, what it does is it packages and ships proteins. If on the whole surface are a bunch of ribosomes, it makes sense that it's responsible for, it'll envelope that protein, it'll envelope that protein and ship it off somewhere. So it'll move some of its membrane away and engulf that protein and ship it off wherever it needs to go. So that's the rough endoplasmic reticulum. So the smooth SER, the smooth endoplasmic reticulum. So what smooth reminds me of, what I also, just as you know, I try to simplify, so we don't need to know all the jobs. Let's just get their main job. So the main job of the smooth ER, it reminds me that it's smooth, is to package and ship lipids. So packages and ships lipids. Okay, so we did SER. It's smooth. Smooth reminds us of fats. It packages and ships fats. Okay. We talked about ribosomes. Let's talk about the Golgi. So Golgi apparatus or Golgi body that's like the Amazon of our cell. So that does all the packaging and shipping of proteins and lipids and everything else. It's our main hub, like our main Amazon is our Golgi. So that does a lot of the packaging and shipping and delivering throughout the cell is our Golgi. Okay, let's talk about the cytoskeleton. What does it do?
Gives, yeah, structure.
Shape, structure, yep. And then what about cytoplasm?
More like chemical reactions.
Yep, awesome. Yep, so it helps, it's like the,
It's the fluid.
maybe for like movement through the fluid, something like that. Yeah, like where things, everything kind of happens in the cytosol and like everything's kind of moving through that way, and it moves through that viscous fluid and the directional movement it should flow through. Okay, and then mitochondria. Have I picked on my husband yet in this class?
You have.
Yes, okay, powerhouse of the cell, that's all he knows, after we've been married for 15 years. So mitochondria is the powerhouse of the cell. That's where ATP is made, and we know now that if the cytoplasmic membrane of the bacteria were to pull away and form a little mini shape, that mitochondria it's what it's shaped like. I won't get into it too crazy right now because we'll talk about it later. But this mitochondria has this inner membrane here and an outer membrane, and this inner membrane has the electron transport chain all over in the mitochondria. And then this is where the hydrogens are pumped into this inner membrane space of the mitochondria. And then there's the ATP synthase that will add the phosphate to the ADP, and now it's pumping out ATP like nobody's business. Yeah.
Is the outer layer of the mitochondria made of peptidoglycan?
No.
Okay.
It's phospholipids. Yes, great. Yeah, that's a good question. But yeah, peptidoglycan, the only place we see that anymore is bacteria. So yeah, good question. Okay. Mitochondria powerhouse, ATP manufacturing. Okay, and then where are we at next? We did ribosomes, centrioles. Does anybody know what the centrioles do?
They help it split.
Yes, awesome, with cell division, perfect. So they like anchor and they move kind of like little magnets where all of the cytoskeleton will pull and attach to the chromosomes and pull it apart. We'll see that in a minute. Golgi, vacuoles. So any kind of vacuole is to like store, I shouldn't say hold, be a little more fancy. Storage. There's protection, digestion, there's like it's almost like a little immune system within the cell. So if the cell has a virus inside of it, then any of the pieces, it can start enclosing those into lysosomes or whatever the little vacuole is inside. So like storage, let's say digestion, stuff like that. Okay, vacuoles, Golgi, blah, blah, blah. Did I forget anything? Yes. Yes. So cytoplasmic membrane. So this is what's tricky about the word cell wall. So we generally say plants have a cell wall, animals don't, right? That's just broadly, plants have a cell wall, animals don't. But plants have a cell wall made of cellulose. It's different than the cell wall of bacteria is made up of peptidoglycan. But generally, well, human cells do not have a cell wall. We call it a cytoplasmic membrane. It's akin to the cytoplasmic membrane of bacteria. And that is a semi-permeable membrane. What that means is it's a little bit permeable. There's stuff can come in, stuff can go out, but it depends on what it is. Our cells are very selective about what they let in and what they let out of the cell. Yeah?
Are plants categorized as eukaryotes since they have nuclei?
Yes. Yeah. Yes. Great question. Yeah. Plants are eukaryotes. Yes. Everything else that's not a bacteria or an archaea or a virus is a eukaryote. Everything else that's living on the planet. Yeah. Seems weird. Okay. So some eukaryotes have flagella. Some of the organisms that we'll look at have flagella. Some human cells, like sperm, have flagella. Chloroplasts, so some eukaryotes have chloroplasts, so they're able to photosynthesize, but we don't talk about the ones that are able to photosynthesize because they're non-pathogenic, so we don't really talk about those. Unless it's like some sort of algae, like toxic algae that people can get from swimming, but we should forget about that. So centrioles, some cells don't have centrioles, but we'll just put them in our yes category. Let's put them in our all. Cell wall, some eukaryotes have those, some don't have a cell wall, like animals versus plants. And then a glycocalyx, so believe it or not, our cells have a glycocalyx layer on the outside. So our cells can secrete those exopolysaccharides so that our cells can also adhere to things. So our cells also have that glycocalyx layer. Some eukaryotes have that, some don't. Yeah?
What's the difference between like the cytoplasmic membrane and then the glycocalyx since they're both like semi-permeable?
So the glycocalyx, what it is just a sugar. So glycocalyx is only sugar, and what it is it's a sugar that's spit out by the cell. So it's like a little sugar layer. And this little sugar layer it's like spit out by the cell, and so that it will adhere to things so that they can stick. So it's just sugars that it spits out of its cytoplasmic membrane. Yeah, cool. Awesome. Okay, so I'm gonna play you guys this video. This is the one I've been really looking forward to showing you all semester. Actually, it's my favorite video. So this is called The Inner Life of a Cell. Has anybody ever seen it? Seen this YouTube video? Yeah? So this was my proteomics professor at Harvard is the one who made this video, and it's like worldwide super famous. His he gets the credit for it, but actually it was his grad students that made it. But it was the first video that they put together that gives us like a computerized inside look of what a cell might look like on the inside. So now that we've kind of talked about some of our organelles and a little bit of our functioning of what goes on inside the cell, this is about to like blow your mind. So I'll play this for you. So Dr. Biol, he's the never seen that before. It tripped me out the first time I saw it. I thought it was just so cool. So it's, oh gosh, I do that every single day. Where are we? So, what you think of the inside of a cell is like, and how it just opens your mind to just the microscopic sense of it, of how, I don't know, when you look at it two-dimensionally, or you think about it two-dimensionally, you think about it from a cartoon, it's just so different of like actually what the world might be like inside of a cell. So there's one of these you can look at on YouTube that is labeled. So I think that one's kind of cool to watch because it shows you like, this is the cytoskeleton, that's a motor protein, that's a vacuole that it's moving, there's the cytoskeleton, and so it labels those things. So I think it's kind of fun to watch. But yeah, it just tripped me out of the inner life of a cell of what that might be like. Okay, so let's go into, so the reason that I split off eukaryotic genetics is because, so how some of the classes are run, the micro classes, is they'll do, this is a bacteria, this is a virus, this is a protozoa, this is a helminth, this is a fungi. They introduce all the organisms at the beginning, and then they'll introduce metabolism and say this is metabolism, and then these fungi can do this kind, these bacteria can do this kind, these eukaryotes can do this kind, and that kind of confuses me. So, when you're talking about metabolism or you're talking about DNA, they work so differently between the different organisms. So I chose to organize the class in this way. So I know we're jump around in the book a little bit, but eukaryotic genetics are different than prokaryotic genetics. And when you're trying to compare a bacteria with a single chromosome, a single circle chromosome, it's arranged into operons. There's not really mRNA in bacteria. There's just RNA. It doesn't have to go through the M process. The genetics are just different. And so even though we shove a bunch of information up front, now you already know what genetics are. You already know how to make a double-stranded DNA. You already know how to make a single-stranded RNA. You already know how to make proteins from that, and you know some of the pieces. So now we're just going to learn the differences. So this is what's different about eukaryotic genetics versus prokaryotic genetics. So here's overall. You have that in the slides, but let me actually go through it, walk you through it. So because eukaryotic eukaryotes are so different, that we all have a different chromosome number. Not we all have the same chromosome number, but organisms within the eukaryotic branch all have different numbers of chromosomes. But does that mean complexity? It doesn't. So like rice, for example, they have 24 chromosomes. How many chromosomes does a human have? 46. So we have 46 chromosomes. So we have 46 chromosomes. We have 23 from our mother and 23 from our father. So we have 46 chromosomes in every single cell in our body, except for 2. What are the 2? Does anybody know? Where is there not 46 chromosomes?
In our eggs and sperm.
Yes, so eggs and sperm have 23 in one cell. They have 23. And then there's one other example, but I don't care that you know that for this class. Just if you guys know it for me, do you guys know what other ones don't have this number or this number? Funky one. Super weird, funky question. So erythrocytes, red blood cells, once they're born, they get rid of their nucleus and they have no nucleus. They have no chromosomes. So just side note, you don't care for this class. We care that there's 46 in humans, there's 23 in eggs and sperm, and we're not gonna go through every organism and how many chromosomes it has. We're just gonna say they have more than one. So bacteria have one, eukaryotes have more than one. So roundworms have 4, but rice has 24. So it doesn't also relate to complexity. So a dog has 78 chromosomes, we have 46. So are dogs more complex? No, I mean, maybe in some ways, but it doesn't always lead to complexity. So we're gonna talk about Giardia, and they have 5. We're gonna talk about pinworms, those have, sometimes they have 1, mostly they have some variation on that. But just so you know, generally eukaryotes have more than 1. And so this is base pair numbers. So remember, a base pair is like if you said A and T, A, T, G, C, C, G, then you'd have T, A, C, G, G, C. This is a base pair. So here in flowering plants have 10 to the 11th base pairs in their genome, where humans, mammals here have about 10 to the 9.5, something like that. Where here's bacteria. So it's not necessarily in complexity either, in the amount of base pairs. So the human genome is this wild ride of all kinds of different stuff, and honestly, we're just learning what the entire human genome does. That was when I went to Cornell, I was 28, and Craig Venter, the one who was kind of the pilot of leading the Human Genome Project, that whole thing kind of had just gotten down, right, when I was an undergrad. So do we know what the human genome does yet? We still have not a clue. We still don't know half of what the human, or 3/4, we probably know 10% of what the human genome actually does. So it's a huge area of research. We're trying to figure out like what all these things do that we always considered was junk DNA. And now we're just getting to know more and more of what it does. So protein coding genes, we think is only about 1.5% of our genome codes for proteins. The rest of our entire genome, we don't know what it does yet. Some of it we know what it does, some of it helps control different things and is organized in a certain way, but a lot of it we don't know. Okay, so let's talk about, the reason that I'm gonna talk about human genetics, because we don't talk about humans in this class, we talk about these guys, protozoa, and we're gonna talk about helminths and fungi and things like that. But the reason I'm gonna talk to you about human chromosomes is because it will help you understand, and we're designed in a similar way. So we have linear chromosomes. So our chromosomes are in a line. They're not in a circle. And one thing I'm going to go through with you when we talk about replication and transcription and translation, that a chromosome looks like this. That's what a human chromosome looks like. What they're always drawn as is they're drawn like this. But that's not a human chromosome. That's a replicated chromosome. So I'll talk more about what that means, but human chromosomes look like this. So we have 46 little pegs, 46 lines. And actually what's inside of there is our double-stranded DNA wrapped and wrapped and wrapped and wrapped and wrapped 100 billion times, wrapped and wrapped and wrapped and wrapped and wrapped and wrapped and wrapped. Then you zoom in again, and it's the double strands. Same as bacteria. It's still double-stranded. It's wrapped around and around and around. I'll show you a better picture of it. And they're wrapped around proteins to make them short and tight, to squeeze them into a tiny, tiny space. So we have 46 chromosomes. So DNA needs to make a copy of itself to get into the next cell, but also it needs to make protein. So it has 2 main jobs. We have to replicate our genome so that we can make another cell, and we have to make proteins. So I said dark bands are AT-rich DNA, but we don't really care about that. It's just something that you look at. When you look at this, this is called what's called a karyotype. A karyotype is like looking at the macroscopic view of the human genome, which it would never be macroscopic. It's microscopic, but it's like a picture view of the chromosomes. So when you're looking at a picture view of the chromosomes, you can see large abnormalities. copies of the chromosome. So a lot of times if you're a geneticist or you're a genetic counselor, you'll start by looking at the karyotype and look at them if there are any abnormalities that are on a macro scale. So here is an XX. So this is the last two chromosomes. So this is what are called sex chromosomes. These are called autosomes, and these are sex chromosomes. Sex chromosomes, if there's an X and an X, it's female. If there's an X and a Y, it's a male. So an X is long and a Y is short. And so this is an X chromosome, this is a Y chromosome. So in a sperm, it will either have an X in it or the sperm will have a Y in it. So in the egg, in a human female egg, always has a human female egg always has a X in it. So when the sperm matches up with the egg, it's the sperm that will provide an X so that it's a female, or the sperm will provide the Y and the offspring is a male. Okay. So here's what supercoiling looks like. So Dr. Myers reminded me, I forgot about that, but actually bacteria don't have histones, they have histone-like proteins. Bacteria have a different method of supercoiling, but this is how eukaryotes do it, is that eukaryotes have these little proteins, these little balls right here that are called histones, and these histones will wrap and wrap and wrap the DNA so tightly so that it will fit into the nucleus. Our DNA is 67 billion miles long, 150,000 round trips to the moon. Totally bananas, right? That doesn't even make sense to me physically. I have no idea how that works. But what it's called, there's another term that I'm going to throw at you, is euchromatin. So euchromatin makes you. That's the chromatin. That's the part of the DNA that's open and ready to either replicate or transcribe or translate. And then there's heterochromatin, which is closed and genetically inactive. So if it's tightly wrapped, then you can't get the polymerases in there. If it's unwound and unwrapped, then you can get the polymerases in there to replicate or transcribe or translate.
Okay, are
Yeah.
you saying that the same sequence of DNA can either be in this form or that form?
Fantastic question.
Or are you saying that one type of DNA is this and one type of DNA is that?
Oh yes, no, it's, so, for example, like the human, so if we have human cells that are like the eye cells, like the retinal cells, then those in the liver, so you have a big liver here, so the genome that makes proteins for the retina is always going to be heterochromatin in the liver.
Okay.
Yeah, does that make sense?
Yeah.
Yeah. So it will wind and unwind if it's going to need access to the polymerases or not.
So just depending on where it is in the body decides whether or not this
Yes, and if it's going to be on or off at any given moment. So
Okay.
it can be on or off with transcription factors also, like the lac and tripoperon, but also it can be heterochromatin versus euchromatin, depending on if it's on or off. It's like all a rhythm of like areas that are open, areas that are closed.
Okay.
So that's one more thing that Dr. Myers brought up that's an important thing to bring up, is that our 46 chromosomes are the same in one little skin cell that comes off, same exact 46 chromosomes that is in my liver, kidneys, heart, eyes, lungs, same exact 46 chromosomes. The reason why they have different jobs, that the proteins can be manufactured from different areas, is because some of it's turned off and some of it's turned on. So like if you look at these chromosomes, then if this little area right here codes for skin color, then it will be like how much melanin is produced from melanocytes or whatever it is. So that will be turned on in the melanocytes that code for skin color, but this area will be turned off in the stomach cells that are going to produce mucus. So parts of the genome are turned on in certain cells, parts of the genome are turned off in certain cells, but they're all the same. Okay, so this is the anatomy of a chromosome. So a chromosome, remember, looks like this, and forever from now on, you know, this is what bugs me about science. Is like, I've taken science for a lot of many years, and I always thought my whole life that a chromosome looks like this. And that's not what a chromosome looks like. That's a replicated chromosome. So I don't know why they draw them like this, always to look like a chromosome. That's not. So this is a chromosome. When it's together in pairs, it's been replicated. That's an exact copy. So let me go through that with you, because that process can be a little bit confusing, but I'll try and make it as clear as I can. So here's one chromosome. Here's what's called a centromere. That's what is like a unit within the chromosome itself. That's the center that where cell division can occur and pull the chromosomes apart. I'll show you what that means. At the end, we have what are called telomeres that are caps on the end of the chromosome. Have you guys heard all the science around telomeres a little bit? So telomeres, they're huge in science right now and probably for the last 10, 20 years, because that's how cells die over time, how we age. There's probably a million things that go into it, but one of the things we know that goes into aging is the telomeres. When the chromosome replicates, get shorter and shorter and shorter and shorter and shorter, shorter, and those telomeres getting shorter, now the replication it'll mess up the replication for the next round, and that cell will die. So then that's why I get wrinkly. That's why my knees start hurting over time. That's why older people get tired, or older people have issues with they're immunocompromised. Their immune system doesn't work as well because those telomeres are getting shorter and shorter every replication round. So as you can imagine, for like longevity research, they're figuring out how to prevent telomere shortening with time. Okay, so replication in eukaryotes happens on a cell cycle. So it happens differently in bacteria than it does in humans. So try and stay with me for a second, because I know it gets to be a lot. But bacteria undergo binary fission, right? We know that they just asexually reproduce, they reproduce constantly. They're always replicating their genome and splitting into another cell, replicating their genome, splitting into another cell. That's asexual reproduction. But the cell cycle is a little bit different. Our cells are on a timer, and they're on a specific timer, so we kind of call that something different. We don't really call that asexual reproduction, and we don't really call it binary fission when we call it the cell cycle. But throwing it out there, with our cells, that's how we do it. But with eukaryotic cells, such as protozoa and fungi, then we do still call it asexual reproduction or binary fission. We use the same term, even though we are now on a cell cycle. These won't just produce. They're born, and then they will asexually reproduce, and then they die. So it's not the same as bacteria that can constantly reproduce with no checkpoints, no stopping points, no other part of their life where they're doing something else. Yeah?
So what you're saying is that the cell cycle for unicellular eukaryotes is called binary fission?
Yes.
Okay.
But it's also called that for bacteria that don't undergo the
Right,
cell
right,
cycle.
but
Yeah, yeah.
but it is called asexual reproduction if it's a one-celled organism, even if it is a eukaryote.
Yes.
But when it's a multicellular organism, the cell cycle is called.
When it's a multicellular organism, the cell cycle is called the cell cycle.
Okay.
Yeah. Yes. I know it's kind of confusing because the terms kind of seem like they coalesce a little bit. But so binary fission. So let's just talk about the cell cycle. So we're going to talk about the cell cycle. So the cell cycle in eukaryotes goes through certain phases and checkpoints. Because otherwise, if it doesn't go through phases and checkpoints, that's called cancer. When cells don't go through those checkpoints of stopping reproducing, stopping the replication process, and going through the checkpoints, and assessing the chemical signals around them if they should reproduce or not, that's what cancer is. So cancer is uncontrolled cell growth where they don't go through these checkpoints. So the checkpoints are G1, S, G2, and then the cell will go through mitosis and cytokinesis. So another thing that bothers me about when you learn about this stuff when you're in middle school, high school, is that usually they'll talk about the cell cycle as if it's mitosis. Like mitosis, this is what mitosis is. Well, mitosis is just a part of the whole process. So the whole process starts when organelles replicate. So the organelles have to replicate and the cell gets bigger physically. Then the DNA will replicate. So that will trigger the next phase. The next phase is called S phase, or the synthesis phase, is when DNA replication happens. So now the genome will duplicate. So just a quick question, in DNA replication, how many chromosomes will we have when it goes through S phase?
46.
92. So at one point, our cells will have 92 chromosomes in them. So that's when they start looking like this. So when people draw this shape and they say that's a chromosome, that's when there's twice the amount of chromosomes in the cell.
But that's
Yeah.
still considered one chromosome, isn't it?
Well, it's still considered one chromosome, but really they are like sister chromatids together.
Yeah, they're duplicates,
Yes.
but as long as they're still attached, they're one.
It's just semantics. It's just, I hate
Okay.
it. But yeah, they should call it a whole nother thing. Or call the single chromatid something different. So yes, it's confusing. There's not another way around it, but sometimes the confusion part will help you understand it. That you just know there's 46, at one point there's 92. That's when it goes through the S phase. So it's gone through the S phase of replication. Now it's gonna go through proofreading, which I'll talk about in a second. Then it's gonna go through PMAT, through mitosis. Prophase, metaphase, anaphase, telophase. I want you to know what PMAT stands for. I want you to know those processes. There's great videos on it. I'll post an Amoeba Sisters one. The only one that we're really gonna care about is metaphase. But we're gonna go through mitosis, prophase, metaphase, anaphase, telophase. What mitosis describes are the chromosomes separating. That's really all it describes, is how chromosomes pull apart. It doesn't describe how the organelles are already, there's twice as many. It doesn't describe how there's already twice as many chromosomes, which in my view is the most important part. Then proofreading, very important. But this only describes how chromosomes come apart. And the only reason that we care about metaphase is because this is a good time to count. So I'll show you that in a minute. And then we go through cytokinesis where the cells split. Now the cells have cytokinesed, there's the splitting that happens. Now one cell was two cells. Okay, so let's talk about replication. So replication in a eukaryote, I know you guys are gonna hate me for this, but now we have new enzymes in a eukaryotic cell. So in a eukaryotic cell, we don't have DNA polymerase 3 anymore. That's bacteria. We don't have DNA polymerase 1 anymore. That's bacteria. We have DNA polymerase delta and DNA polymerase epsilon. But the reason I really like how the scientists used the Greek letter delta and the Greek letter epsilon is because it kind of tells us what it does. Even though this is a funky little S, and it's not really an S, it's a Greek delta, it tells us that's synthesis. Yeah? Synthesis, DNA synthesis. DNA polymerase epsilon, even though that's not an E, it's an epsilon, that tells us it's the editor. That's the proofreader. So we now in eukaryotic genetics, we're not talking about pol 3 and pol 1 anymore. Pol delta is the actual polymerase, the one that synthesizes. And now we have epsilon that does the editing. Yeah.
I feel like in all of my other biology classes, for eukaryotic cells, it's DNA polymerase 3, DNA polymerase 1.
Yeah, that's not really right. Yeah, it's pol delta, pol epsilon are the polymerases. Yeah.
So these overall phases, the G1, S, and G2, stand for something meaningful?
I don't know. Gap phase. It is gap 1, gap 2. Yeah.
And is the S?
And S is synthesis.
Okay.
Yes. Yeah.
So the DNA polymerase,
Delta,
no,
epsilon.
yes.
Uh-huh.
Is like the secondary,
Yeah.
polymerizing enzyme, so it goes in after and proofreads.
Yes,
Okay.
that goes through G2 and that does all the proofreading. And if we don't have a good proofreader, that's when we have mutations. Yeah? Okay. So
And
we
all eukaryotic cells have both
all eukaryotic cells have both.
that we
Yes.
care about.
Yeah. Yes. So there are many origins of replication. So in a bacteria, remember we had one circular chromosome, one origin of replication. In eukaryotic chromosomes, we have hundreds of origins of replications. There's not 46, there's not 23, there's hundreds of origins of replication. Usually in eukaryotic genetics, we call those a data box, but don't know that. Just know there's many ories. Okay, replication happens in the same way. It happens, so here's DNA replication. These are called homologous chromosomes that are, we'll talk about that later. So replication happens in the same way. Those are the only real differences that we need to know, is that they have different polymerases, but they have, this enzymes work in the very same way, and we have almost the same enzyme. So remember we had a different topoisomerase, but I don't care if you know that we have basically the same enzymes and it works in the same way. So same semi-conservative replication where the DNA polymerase S, delta, will come through and make a new copy. So now we have semi-conservative replication. We have mitosis. This is prophase, metaphase, anaphase, telophase. Here is a really good Amoeba Sisters video on that talks about chromosome number. So it talks about how when we replicate, we have 92 total chromosomes, and those 92 will split off, and then you'll have 46 and 46. So you can watch that on your own. So the last thing I'm going to mention, let me see how far we got. Oh yeah, we're almost done. Okay, so one more thing that we're going to mention is, so chromosome number, really quickly. I just want to make sure that you can do this on your own. So on your own, and I'll do this at 12:30 on Tuesday. Next Tuesday, I'll do this again at 12:30. Is if you have an organism with 4 chromosomes, and it's going to go through replication, it's going to go through S phase. So now the chromosomes are replicated. So now we can say there's 8. But like Dr. Meyer said, really they still say 4, but just forget that. You're just gonna do my way, not every other scientist's way in the whole wide world. So there's 4 chromosomes, there's 8 here. Now they line up on the metaphase plate. So remember I said this metaphase is the only one we care about. So if I ask you on an exam or a quiz, how many chromosomes, or like there's a organism that has 5 chromosomes. Draw this organism in metaphase. Then you'll know it has to have the double amount because it's already gone through the S phase. It has to have twice the number here. And so now here it has 8 chromosomes lined up on the metaphase plate. Those are going to be pulled apart by the centrioles. And then now we have 4 in one cell and 4 in another cell. Does that make sense? Yeah, why I care about the metaphase plate and why then they split off and now there's 4 in each cell. So that's replication. Yeah.
Are we gonna be studying meiosis in this class?
Yes, but I try and do meiosis separate because then we're talking about sexually reproducing organisms.
Mhm.
So yeah, and then hopefully you like master mitosis, and then meiosis can come along later. Hopefully, yeah.
So if you ask us how many chromosomes are in that metaphase picture right there, we should say 8.
Yeah, but I won't ask you the number of chromosomes because some people say there's still 4, even though there is 8. But I'll ask you if you have an organism with 4 chromosomes, draw metaphase of the mitosis. Then you're expected to double the number and draw them along the line.
Okay, so if it asks us to draw 4, then we should draw 8.
Yes, if it's on the metaphase plate, because already it's gone through here. So see how there's 4 greens, and then there's 4 greens, and then there's 4 reds. So this is 1, 2, 3, 4, 5, 6, 7, 8. They're together at the centromere. Does that make sense now, or no? Okay, let me explain it again. So here, if you have an organism that has 4 chromosomes, when it replicates, they're bound together at the centromere. So here's the centromeres. The DNA pole delta went through and replicated. So now, one of these guys just looks like this now. So it went from looking like that to looking like that. So they're just tied together at the centromere. So here's number 1, here's number 2, here's number 3, here's number 4. Here's number 1, 2, 3, 4. But it copied, so here's 5, 6, 7, 8.
Okay, so it's S phase, and then double.
Yep, and so here's 1, 2, 3, 4, 5, 6, 7, 8. Yeah?
What's the reason for the arrow coming from mitosis?
Well, it really, yeah, no, it really should be replication,
Yeah.
should be here. This is replication, and then mitosis should be here.
Yeah.
This is metaphase of
So the
mitosis.
first one is your G1, right?
So this is, well, G1 is, yeah, where all the organelles are copying. Yeah, I just wanted to show you what happens at S phase and show you what happens at metaphase and cytokinesis. Yeah,
Well, we didn't need to worry about like those spindle fibers.
no, I want you to watch it just so you know what prophase, metaphase, anaphase, telophase looks like. But that's just semantics. It's just pulling the cells apart, pulling the chromosomes apart.
So we
The
don't need to
biggest
draw
thing
the
is
central-
metaphase, because that's how they're lined up. And then S phase, that's how they've replicated. Yeah,
So when we draw it, we don't need to have centrioles and stuff?
no,
Okay.
this is all I care about, chromosome number. Yeah, okay. So then they split apart, this one has 4, this one has 4, just like the mother cell. They do that, call those mother cells daughter cells. These are daughter cells, that's the mother cell. Yeah,
Sorry, just to clarify.
No, please, anything.
In
Keep
S
going.
phase, we would say there's 8 chromosomes there.
there are really 8 chromosomes, there are.
Okay.
But some scientists will still say there's 4, even though they're double the number.
Okay.
Yes. But I will say there's 8. Okay, okay. Okay, so you guys ready? Move on? Okay, I'm gonna move on to this next part and then we'll get going. So another difference between eukaryotes and prokaryotes is in transcription and a little bit in translation, but nothing too crazy. I'm gonna erase this. Everybody okay with that? Yeah? Okay. Okay, so in Eukaryotes, what happens is we still have RNA polymerase. We don't have to learn a new one. We still call it RNA polymerase. RNA polymerase will bind to the promoter and transcribe and translate. In eukaryotes, we don't really call it an operon anymore because there aren't coordinated genes into an operon like there are bacteria. But let's just take it on. It does bind to the promoter region. Here's the promoter. And the RNA polymerase will make RNA. So now what's going on with this RNA? Let me stop there. There's a piece of RNA, just like always. But this RNA, now it goes through another process that bacteria don't. So we have introns and exons, where bacteria don't have that. So introns help me remember that they're in the trash-ons. Those ones go in the trash. Exons code for proteins, amino acids. So that's how I've always remembered it. Introns go in the trash, exons are the ones we need. So it goes through this process called splicing. So it'll splice with something called a spliceosome. I hope that's how you spell it. A spliceosome. And that spliceosome, what it does is it takes those intron pieces. I don't have black anymore, so I got it. But it cuts them out. It'll form these little rings and then adhere these two sides together and it will cut them out. So our resulting RNA strand will look like this. It'll be shorter, right? Because we took those 3 exons and put it into one piece of RNA and now it's been spliced. Yeah.
Where does splicing happen?
In the nucleus. Yeah, so we'll talk about that. So splicing happens in the nucleus. Now what happens is it will gain a 5' cap, which is like a methyl group and something else. Just call it a 5' cap. It'll gain some, it's like a methyl group with something else. I can't actually remember off the top of my head, but it's a 5' cap and then a poly A tail. So that's the 3' poly A tail. And so that's, tail. So that's a adenosine, or adenine. It adds the adenines to the end. So now this is called mRNA. So sometimes you'll hear bacterial RNA called mRNA. A lot of people just call mRNA. But really, once it's matured, we can call it mature RNA, or sometimes it's called messenger RNA, depending on who you ask what that M stands for. So mature or messenger RNA, it's been spliced, a 5' cap has been added, a poly A tail has been added. Now it can leave the nucleus. Now that it leaves the nucleus, it goes through translation in the cytoplasm. Now the ribosome, ADS ribosome, will come along and read it, produce an amino acid sequence that produces a protein, in the same manner ours does. Bacteria does. Yeah?
Can you just remind me again of what the ADS is composed of? Is it 60 and 40?
60 and 40. Yes. Awesome. So here's the exons, here's the introns, here's the 5' cap poly A tail. Oh, here's the methyl, guanine, phosphate, or if that's phosphorus without the phosphate, I can't remember. But here's between 150 to 250 adenines. Tons of adenines at the end to protect it. We have a cap and a tail, and it's been spliced. So here now it's out of the cap, the tail has been spliced, it leaves the nucleus. Here's comparing bacteria to eukaryotes. Okay, So here are the terms. We talked about some of these terms. I can go through those again one more time, but let's go ahead and do our note card. Okay, so actually we're sticking on our one last virus, and then we're gonna move on to our eukaryote. So this is our last virus. So we've talked about it already a little bit. This is influenza virus that causes the flu. No, you're okay with everybody not turning them in? Yeah?
Yeah.
Okay, cool.
I've taken it.
Got it. Okay, perfect. So here's influenza virus. Now we know what these are. We know the capsids. What type of capsids are those shaped in? Polyhedral capsids. It has an envelope, so it's an enveloped virus. It has spike proteins. Did I say human influenza virus? I hope I didn't say that. It's just influenza virus. Never let me say human in front of it. Even though this says human, but depending on, it can be birds, it can be other animals too. So it's just influenza virus broadly. Okay, so it's negative sense single-stranded RNA. I don't care that you know it's segmented, but just so you know, it has a lot of different little negative sense RNA strands. It's not one big long one, it's separated into different ones. It's enveloped, it has hemagglutinin and neuraminidase are the H and N. So now we know what H and N do. H helps it get into the cell, and N helps it be released from the cell. Yeah. Oh yeah, so let's talk about that before we get to the final. But our RNA is here, it's our 5' cap and our poly A tail. And this is the 3' end. Well, it'll be like a 5' A and a 3' cap. So the RNA or the ribosomes will start over here. The ribosome would start here and fall off the end. Ribosomes will only go they can't go that way. They can't go backwards. It wants to go that way. So what happens is it comes with a replicase, or whatever it happens to come with, and that will reverse transcribe it. So then now it has the 5' cap and the poly A tail, and now our ribosomes will start at the beginning and go all the way to the end. It's not really that important for you to understand it, just know there is a negative sense, it has to be in positive. Positive is a positive thing, we need positive. Positive is what our cells use. That's all you need to know. Okay, so respiratory, also influenza virus can cause meningitis also. There's 16 different versions of hyaluronidase that we know of. There are 9 versions of neuraminidase that we know of. It's not a provirus, it's not an oncovirus. There's A, B, C, D types. A and B are the most important. So zoonotic diseases, we worry about that. Influenza, between about 50,000 deaths a year on average, usually 30 to 50. Hospitalization, 700,000 a year. Illnesses, 41 million a year. And then here is our zoonotic diseases. So here's the different H1N2, here are the different years that those have come about. H1N1, H1N2. Here's just some little more information. I think that's really interesting to know. And then remind me, we're going to start talking about zoonotic one more time. Yeah?
Can I see the picture of influenza?
Oh, yes.
please?
This one or this one?
That one.
Okay.
Thank you.
Is there anything we need to know about prions for the final?
No, if I have like 10 extra minutes in a lecture somewhere, I'll throw it in there. If not, we won't. Okay.
Will we need no spelling for the final?
No, I don't usually take off spelling unless I really don't think that you know what it is. You know, like some people put capsule instead of capsid, so those are two different things. So just make sure that if the spelling is wrong, that I at least know what you're talking about, and spelling I don't care.